So formative assessment 5.1, reviewing main ideas. During which stage of the cell cycle is the DNA copied? During which stage is the DNA copied? So the answer here is during the S stage. During the synthesis or S stage. So for such a question, it's just a matter of you having a good memory. So let's look at the next question. That is question 2. Question 2 asks, which stages of the cell cycle generally requires about the same amount of time in all human cells? In all human cells. So actually the G1 phase, G1 stage will vary depending on cell to cell. Even the G2 may vary depending on the needs and even the age of the human. But the one which doesn't vary is actually the synthesis. So the synthesis phase or the S stage will take the same time in all the cells. This is actually during DNA replication. So when the DNA is being duplicated, it takes actually the same time. Let's go to the next question. What limits the maximum size of a cell? What limits the maximum size of a cell? What limits the maximum size of a cell? The maximum size of the cell is due to the ratio of the cell due to the ratio of the cell surface area to volume. So the reason as to why it's like this is that the cell actually needs to transport materials in and out of this and remove materials out. So oxygen and nutrients and waste move across the cell membrane or the surface of the cell. Actually, movement of materials in and out of the cell is affected by the cell's surface area. So if the surface area to volume is large, then it will be very difficult to move materials within the cell, and the cell may not function as required. So materials must be transported. Materials must be transported at at adequate speed. At adequate speed to keep the cell functioning. So if materials, for example, will delay to reach where they are required to provide energy, or if the wastes will overstay in the cell, then it will be a disaster for the cell. Next question, we got the next question. Question number 4. We go to question number 4. Question number 4 reads, this is about thinking critically. Infer. Suppose you were to draw a diagram representing the cell cycle of a neuron. Explain where and how you would represent the G0. Explain where and how you would represent the G0. So let me bring a diagram of the cell cycle so that it's easier for us to see what we are meaning here. So actually when a cell is dividing, it will go through interphase, then it will enter mitosis, then cytokinesis. After this, the cell is already divided, and when we are talking of G0, we are saying that the cell will enter a cycle where it will not divide. So I would expect, or we will expect, that the G0 is actually in between here, in between or after cytokinesis. So after cytokinesis, we'll have our G0. So it will stay here in the G0 unless or until when the conditions would allow it to go through the cell cycle again. So we are meaning here the cell would go straight to the G0 line. So you would represent it in the cell cycle after cytokinesis. Because at this time the cell is already or has already completed has already completed the cycle and is ready for a new cycle and is ready for a new cycle. For a new cycle. So it would be ready for a new cycle, but because this cell is not supposed to divide immediately, it will instead enter the G0, and in the G0 there will be no division until when division is needed. So let's go to the next question. The next question here asks, make a prediction. Suppose you treat cells with chemicals that block cytokinesis. Describe what you think the cells would look like. Describe what you think the cells would look like. So as we were discussing, we looked at both mitosis and cytokinesis. Mitosis is the division of the nucleus. 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 And therefore, what we are meaning here is that something that will actually affect mitosis will also affect cytokinesis. So we will say here that cytokinesis, we will say that cytokinesis, we'll say that cytokinesis is the division of the cytoplasm. Cytokinesis is the division of cytoplasm. So what happens is that if cytoplasm is actually not divided, if cytoplasm is not divided, then what happens? If cytoplasm is not divided, then what would happen is that the cell will have two nuclei. So the cell will have two nuclei, and apart from it having two nuclei, it will also have twice the DNA. It will contain twice the DNA because cytokinesis was stopped. So if cytokinesis occurred without mitosis, if mitosis occurred without cytokinesis, the cell would contain two nuclei and twice the DNA. The reason being that the cell will not enter the division phase. Since the cytoplasm will not divide, the number of DNA will be duplicate, and even the nucleus would also be a duplicate. So we'll stop here for now. So as we said, if it happens that cytokinesis, which is the division of the cell cytoplasm, stops, then and mitosis occurred without, if mitosis will occur without cytokinesis, then the cell would contain two DORA nuclei and twice the DNA. So this is the answer. Suppose you treat a cell with chemicals that block cytokinesis. So what will happen is that if mitosis occurs without cytokinesis, this one is supposed to be actually cytokinesis. Then the cell would contain two nuclei and twice the DNA. If cytokinesis occurred, on the other hand, without mitosis, one of the new cells would lack DNA and the nucleus all together. So we'll stop here. Hope you are benefiting from these videos.